Welcome to Chapter 20 of Microbiology. Chapter 20 needs to be approached with a bit of humor. We're going to talk quite a lot about feces, fecal-oral transmission, and of course, all the lovely symptoms that patients come down with as a consequence of fecal outbreaks. And so our gram-negative bacteria have a wide range of habitats. They are located in animals being zoonotic, some of which do persist in our respiratory tracts. And then, of course, the ones that are going to be responsible for gastroenteritis are most often obtained from soil and water. All gram-negative bacteria have lipopolysaccharide on the outer portion of their cell wall. As a reminder, lipopolysaccharide functions also as an endotoxin. Pseudomonas aeruginosa is an aerobic bacteria that is gram-negative. So Pseudomonas aeruginosa is often picked up by soil and water, but most recently it's been transmitted in hospital settings. In particular, it is resistant to soaps, dyes, quaternary ammonia compounds, and disinfectants, so things that we would use to clean a hospital room. It's resistant to drugs, drying out, so clearly this is a microbe that could cause a bit of a catastrophe if found in a hospital setting. So it tends to be a frequent contaminant of ventilators. There are instances where they have found it in IV solutions or anesthesia equipment. The microbe overall is an opportunistic pathogen. Small amounts of it are located on the body, and so if these equipment are not properly cleaned or sterilized between patients, that is how this microbe is disseminated. In particular, if this microbe reaches people who are immunocompromised, such as burn patients, those suffering from neoplastic disease, and especially children who are suffering from cystic fibrosis, a lung disease, there's definitely the risk of this complicated pneumonia. And so I've had colleagues, I've had friends who studied Pseudomonas aeruginosa, particularly in the lung, and correlations with cystic fibrosis are very high and can actually be very deadly. And so cystic fibrosis is a comorbidity that would contribute to the severity or lethality of Pseudomonas aeruginosa. Endocarditis, meningitis, and bronchopneumonia. So these are systemic effects of Pseudomonas infection. So if the bacteria leaves the lungs and becomes a systemic infection, the Pseudomonas aeruginosa bacteria on a regular nutrient agar plate by this greenish blue pigment, it's a piocyanin. So they produce piocyanins when they reach proper thresholds or high densities on plates. And it's interesting because this is probably one of the few bacteria that you would work with in a microbe lab that does not stink. So it actually smells very fruity. It has a great black odor. One of the indicators of patients who have severe Pseudomonas infections in their lungs, their breath will also smell fruity. Because this bacteria is multi-drug resistant, it's a huge concern for hospital settings and making sure that you've got proper PPE and don't disseminate this to other patients under your care. So the main reservoir for Pseudomonas aeruginosa is soil and water, infected individuals, asymptomatic carriers, wild animals. Best choice is soil and water. Other gram-negative aerobic rods are Atenobacter and Stenotrophomonas. Both of these are very similar to Pseudomonas. However, they are obligate aerobes. They don't ferment sugars and they're opportunistic. Atenobacter baumannii causes pretty severe nosocomial infections. It happens in wounds, urinary tracts, burns, and blood. It is extremely resistant to most antimicrobials and often has to be treated with a combination of them to be completely eradicated. It is the most recent form is carbapenem resistant. So broad spectrum means that this antibiotic works well on gram-positive and gram-negative bacteria. The beta-lactam antibiotic is the same family of which penicillin is derived from. And so this broad-spectrum beta-lactam antibiotic is inactivated because this bacteria forms the carbapenemase. It's an enzyme that will digest and cleave the carbapenem antibiotic. And so you can see here impenemum, which is a carbapenem antibiotic located in the MRSA case study that you did in class. Atenobacter baumannii has exploding with resistance to impenemum. Stenotrophomonas. You may recognize this bacteria name from the homework video, Hunting the Nightmare Bacteria, because it caused a pretty severe infection in the gentleman who was traveling to India. And so this nosocomial infection can be hospital-acquired or community-acquired. In the case of the video, it was indeed a community-acquired infection. Because Stenotrophomonas can potentially become a contaminant of disinfectants. In particular, it can grow in our antimicrobial soap. So if you are a nurse and you're going in to see a patient and you wash your hands before entering the room, you wash your hands after, if this is contaminated with Stenotrophomonas, you're just coating yourself in this bacteria and then walking into a patient's room. And often, you then are acting as a passive carrier or potentially becoming an active carrier of disease because you yourself become infected with Stenotrophomonas. And so because this bacteria is highly drug resistant, we found that it can be isolated from various tissues in patients, and it's a huge concern for individuals as they work in the clinic and trying to reduce the spread of this between all other types of sources, and of course facilitate the reduction of Stenotrophomonas in our population. Francisella tularensis. This is the causative agent of tularemia. So rabbit fever, you can see a particularly large swollen lymph node in the right corner here. And so this patient is suffering from a pretty severe form of tularemia. The transmission of this can be spread by rabbits, ticks, infected animals. And so here in Missouri between 2000 and 2007, most predominantly it's transmitted by tick bite. And so you can see that the cases are more prevalent in warm seasons. The death rate is nearly that of 10% if you have both systemic or pulmonic forms, depending. In the case of Francisella, this bacteria escapes by hiding out in macrophages, similar to that of Listeria monocytogenes. There is a attenuated vaccine, so a live attenuated vaccine has a modified form of Francisella tularensis, so the microbe has had certain virulence factors removed, can replicate in the body, but does not cause harm or severe disease, such as it does in the systemic or pulmonic forms. Bordetella pertussis. This is the causative agent of whooping cough. It at one point was highly communicable, but due to vaccination efforts, this has been pretty reduced within our populations here in the United States. The reservoir was healthy carriers, and so contact was transmission of direct aerosol droplets or inhaling of aerosol materials. And so because the patient would be constantly coughing, the likelihood of having direct contact or inhalation of aerosols is much, much higher with this type of communicable disease. It was more prevalent among children. And so you can see here that whooping cough in 1976 versus 2010, it was increasing, and then in 2006 there was a bit of a dip, and then it kind of went back up. And so take a moment and try to postulate why whooping cough would have had this dip and then increase in the epidemiology chart. So with this sort of evidence, you could potentially go online. Often in my on-ground classes, I have students do a bit of a debate. I allow them to do a bit of research online and figure out, okay, so why would whooping cough increase and decrease? And in this case, probably my best argument that I have heard, again, it's somewhat circumstantial and it's more of a hypothesis, is that the vaccine for whooping cough actually changed. And so they were using a vaccine that was released, and then this vaccine actually was changed, and then of course it went down, and then it's going back up. So there's certainly some efficacy issues. And then overall, when we think about the vaccine, I think there's the DTaP and the Tdap and, and the alternated versions of these vaccines. Not all of them are effective. And in the case of Bordetella pertussis, the vaccine is really targeting the toxin because the toxin is why you have the whooping cough in the first place. The toxin binds to the ciliary cells, and then the cilia then is paralyzed. With the inability of the cilia to move mucus along, then the cilia is non-functional, is completely destroyed, and the patient then cannot move mucus out of their lungs, which perpetuates the whooping cough, which, you know, is how the bacteria has gotten its name. Legionella pneumophila is the causative agent of legionnosis. So legionnosis has a close association with amoebas. It is distributed throughout water sources. So you can see here, these are Legionella bacteria growing and living symbiotically in an amoeboid cell. The Legionella pneumophila caused a pretty significant outbreak in 1976. This outbreak affected quite a few Legionnaire members, and it was due to a contaminated hotel air conditioner. And so if you're interested, you can click this link for more information. Legionella is not communicable from person to person, but instead is communicable when you come in direct contact with contaminated sources. So hot tubs, contaminated air conditioners, decorative fountains, and so you have to actually breathe in or consume components of the contaminated water. More often than not, the infections take place in elderly individuals. So the nosocomial disease has been reported distinctly in males over 50. And then the Legionnaire's disease, this is the most severe form. It is characterized by primarily pneumonia. And then Pontiac fever is, of course, the milder form. And it's typically going to completely resolve as an acute upper respiratory infection that's, you know, only going to last for a few days. In the more severe forms of Legionnaire's disease, fatality is up to 30% depending. And the contributors for fatality rates to be so high is more so that you have patients who are immunocompromised or elderly, and then those populations are going to have a much higher fatality rate than your average person. The Enterobacteriaceae family, so this entire family breaks down the remaining bacteria that we'll discuss for this lecture. And so there are primary pathogens and then opportunistic pathogens. Shigella, Salmonella, and Yersinia are all primary pathogens. They're going to cause disease regardless of human health. And then opportunistic pathogens are E. coli and Klebsiella pneumoniae. And so these guys are located in all sorts of locations, similar to the previous gram-negatives that we were talking about. Enterotoxins are going to be responsible for causing intestinal damage and diarrhea. And then pretty much when we combine all of our enteric bacteria and Pseudomonas, this is pretty much the half of all nosocomial infections that you're going to deal with in the clinic. So this is pretty much a lot of bang for your buck in a single lecture. So this overall family, if you were to try to isolate them in a lab, you would want to grow them in aerobic conditions, but you could distinctly push them out into anaerobic conditions because they are facultative anaerobe. And just as a bit of a reminder, facultative anaerobes mean that this is a microbe that is normally aerobic, grows best in air, but it can grow in the absence of air. So it can be anaerobic when needed, or at least for short periods of time. These bacteria ferment glucose. They're oxidative negative, catalase positive. These are a few tests that you have probably experienced at some point in lab, or you may want to look up and get some information on. We divide all of our Enterobacteriaceae into coliform and non-coliform. And so coliforms are bacteria who ferment lactose. You can see here this is a MacConkey agar plate. So this MacConkey agar plate has things like crystal violet in it that can be picked up, and if the bacteria ferments lactose, it'll turn a purple pigment or a pigmented tone, versus bacteria that do not ferment lactose will remain somewhat clear, colorless. And so we can isolate various enterobacteria by doing very selective and differential tests. Selective tests are tests that will only allow the growth of one type of bacteria. An example of that is MacConkey agar. MacConkey will only allow gram-negative bacteria to grow. And so it inhibits the growth of gram-positives. Likewise, EMB is another selective agar that will only allow gram-negative bacteria to grow. And so if you had a mixed sample and you put that mixed sample on this plate, you could guarantee that anything that has colonies on an EMB or a MacConkey plate are going to be gram-negative. So you could confirm that with a gram stain, but then you could look at these individual mixed colonies, pull those off, and then do differential tests to distinguish them from their plate mates or what have you. And so some of the differential tests that we would do, so differential tests allow you to distinguish one bacteria from others. And these differential tests allow us to distinguish things like E. coli versus Serratia, which is really of absolutely no harm, versus things like Providentia and Pseudomonas. And so the V here shows that this is a variable test result depending on the distinct species. But overall, in lab, more often than not, our students do go through and they'll isolate these bacteria and they'll run tests, and by the end of the lab, they can figure out exactly which bacteria they have based on the differential results. So for example, E. coli versus Citrovector, if you were just comparing these two, you could see that the differential test is that of citrate. Citrobacter is going to be positive for citrate versus the E. coli will be negative for citrate, meaning that they are differentially determined based on the outcome of the citrate test. Enteric bacteria often have a lot of variance factors. The fact that they cause nearly 50% of all mucosal infections is because they have lots of ways and mechanisms to do those sorts of infections. They possess flagella, capsules, or fimbriae. They have distinct cell walls and, of course, endotoxins in the presence of lipopolysaccharide, and then they secrete exotoxins, which are highly specific for their target cell. And so when we talk about things like Escherichia coli or E. coli, it has distinct capsules that allow you to distinguish one strain from the next. For example, the hemorrhagic strain is E. coli 0157H7. So the 0157 is an O, somatic or cell wall antigen, versus the H is talking about the flagella. And so it's the flagella that was ranked at 7, so 0157 is the 0157 strain. And so we characterize our virulent strains of these types of bacteria based on their virulence factors or surface antigens. Which of the following is true of the enterics? Some are normal flora. Some release endotoxin, some release enterotoxins, or all of the above. Best answer is all of the above, because we do have some enteric bacteria. Some strains of E. coli are completely normal flora. Additionally, because they are gram-negative, they all have lipopolysaccharide in the outer cell wall, and as a consequence, then they possess the endotoxin if the bacterial cell wall is released. And then enterotoxins, some produce enterotoxins, which is true in the case that bacteria secrete enterotoxins causing diarrhea. The transmission of fecally acquired bacteria often results from contamination from a fecally infected host, and it can transmit to the 4 F's. So the 4 F's include fingers, flies, fields, and fluids. So fingers, flies, fields, and fluids, and then that coordinates itself and congregates at food. So for example, if we happen to we go to the bathroom and we don't properly wash our hands, then we go to eat food, we can actually infect ourselves with normal commensal flora just simply by the infectious dose. Bacteria that get to locations they don't belong cause infection. Flies are in close association with feces, and so flies coming on food and things like that actually can transmit bacteria from a fecal source to plates in the food. So this can happen, say, for example, at picnics in the summer. Fields, the drain off of fecal content from both humans if you have an area of poor sanitation, or the drain off of fecal content from infected cattle or CAFOs, for example, so concentrated animal feeding operations. So CAFOs are notorious for draining into the localized fields in that area, and as a consequence, the local crops then can get contaminated with feces from animals that may have been, infected with a specific strain of bacteria. And then, of course, the drain off from poor sanitation or those CAFOs can reach our water supply. And if it reaches the water supply, you then directly may pick up fish or other food sources from that water supply and ingest those. And so it is a vicious cycle. There are ways to prevent it that we'll discuss at the end of the chapter. But there's quite a few outbreaks. This is not a third world issue. This happens on several times, probably each time I teach microbiology. There are distinct outbreaks, and often I'll feature them in homework assignments or discussions. And it's ultimately you've got to cook your food to proper temperatures. You've got to wash your hands. You've got to make sure that you wash your food. So if you're getting beautiful green spinach, but you don't wash the green spinach, there is a risk that it could be fecally contaminated. And so pre-washed foods are always, to me, a point of concern, because you pay for them to be pre-washed, but then ultimately they're probably not as good as you washing them at home, because you're one person and you're washing in one sink versus an entire manufacturing process that are trying to clean green vegetables or spinach or what have you and make it safe for consumers. The efficacy is just not the same as what you can achieve at home in your own kitchen just by spending a little extra time underneath the faucet. So Escherichia coli is the prototypical poo bacteria. There are over 150 strains. There's both aerobic versions. There's quite a few non-fastidious versions, meaning they're not picky. They can grow in just about anything, as well as survive complete saturation in water. These gram-negative bacteria are coliforms, so they ferment lactose. We will do coliform counts to determine the fecal contamination in water supplies. So if you own your home or your parents own their home, you typically within that community should get a water report annually. Sometimes you receive those a bit more often. And those water reports should tell you how much fecal contamination were in your natural water supply, so the water coming from your tap. And because of the fecal contamination in those water supplies, we find that if we travel, Escherichia coli, certain strains of that cause nearly 70% of all travelers' diarrhea. And so when I usually make the joke that, well, when traveling to Mexico, you should probably drink the tequila or the Corona. But ultimately you should drink a packaged product, something that has been sterilized. Bottled water is equally as favorable, but tap, because of sanitation being very different from what we have here in the United States, it could be a bit risky, and you may suffer a bit of Montezuma's revenge. But ultimately, E. coli is not a bad bacteria. It's a bad bacteria when it gets into a body that is not experienced or exposed to that bacteria on a normal basis. So people who live in these climates, their body are used to the type of E. coli, so they can drink the tap water and they have no ill effects. It is, when you're traveling and you are not from that area, your body has not acclimated to exposure to that level of microbe, and then you can unfortunately have a bit of a ruined vacation. And so Escherichia coli, because of being a fecal bacteria, it also causes, is the most predominant cause of UTI. So, the distance between the anus and the urethra are so short that you need to be able to, do everything you possibly can to keep those areas separate. Don't allow E. coli to get into the urethra so that UTIs do not take place. The pathogenic strains of E. coli, so enterotoxigenic, this is the culprit of our traveler's diarrhea, and it's because of a heat label toxin that this bacteria produces. It can take place within 5 to 7 days, so it's not an immediate action, but some cases, depending on the infectious dose, could happen sooner. There is an enteroinvasive E. coli, so this is going to cause a pretty nasty inflammatory disease. So lots of inflammation in the intestine. And of course, this is going to be similar to things like Shigella or a dysentery. Enteropathogenic E. coli, this is very poorly understood. So infantile diarrheas are often caused by enteropathogenic E. coli. We don't fully understand why certain infants have a higher prevalence or illness with this sort of disease, and it's still an active area of research. Now, the most commonly studied and the most probably prevalently known is that of the hemorrhagic E. coli. Hemorrhagic E. coli, the prototypical strain I'd mentioned before, is E. coli 0157H7. And so this prototypical strain produces Shiga toxin. So Shiga toxin is the same toxin that is going to later be produced by Shigella. And that Shiga toxin causes quite a lot of damage in the gastrointestinal tract. Because of the hemorrhagic syndrome, where the red blood cells are lysing, so it's hemolytic uremic syndrome, the lysis or rapid lysis of those red blood cells can cause kidney damage. So the red blood cells, if after they have burst, have to be filtered out and secreted. And so the kidneys can get blocked up and damaged trying to process the massive quantity of red blood cells that have died. And then this tends to be more prevalent in children simply because the immune system is more permissible. It happens pretty rapidly, so 2 to 14 days is the overall expanded window. And of course, this guy is pretty virulent. It's only going to take about 100 cells to cause disease, and that's very few. That's probably enough cells that could be on the end of a needle, for example. So the pathogenesis of E. coli is transmitted from an infected host. So it could be animals that are in a CAFO. You have one animal that is contaminated, other animals are eating food, they pick up the feces. As a consequence, the bacteria then starts to colonize the animal's gut. The bacteria can either go and attach to the microbial surface of the intestine, so the microvilli, or it could try to penetrate and go systemic, passing through the mucosa. And so once the bacteria has passed through the mucosa into the blood, complement proteins are going to opsonize and generate membrane attack complexes to try to lyse this bacterial cell. Phagocytes are going to come and try to phagocytose and clear out the microbe. But once the microbe has reached the systemic stage, or this lower area near the blood vessels, it can produce toxins. Those toxins can cause a really, really nasty effect. So it can cause localized diarrhea, but some of those toxins can go on and produce septic shock and death. And so opportunistic coliforms, such as Klebsiella pneumoniae, this guy is a normal inhabitant of the respiratory tract. Again, just as a quick reminder, coliform means that it can ferment lactose. So you can see here, this is an EMB plate. This bacteria is turning pink because it can ferment lactose. This bacteria produces a very large capsule, which is necessary to protect it as it enters in the respiratory tract. Can cause things like nosocomial pneumonias, meningitis, bacteremia. But overall, it's normally an inhabitant as long as your immune system is intact. It does not cause diseases. Klebsiella pneumoniae also exists in small quantities in the GI tract, so it can be a culprit for various wound infections if people aren't washing their hands properly, or of course UTIs. Other opportunistic coliforms are Enterobacter, Citrobacter, and Serratia marcescens. I leave these on the slide really strictly because I work with them in the on-ground lab. And so it's just always nice to see that the bacteria that you're working with, it's not terribly deadly, but can cause disease and does require a bit of respect. And so that is really why they're here. They do cause opportunistic infections, so Citrobacter can cause UTIs because it's in the GI tract. If it were to spread, it causes bacteremia. Serratia, this guy's really only a concern if you're severely immunocompromised, so burn victim. If you've got a deep wound infection with Serratia, then you could be concerned. But it's so mild that there was a period in time where people would actually study the movement of microbes by using this microbe. They release it in air vents and such, so it's pretty safe. But it does create these beautiful brick red colonies on plates, so it's fun to use in lab because, you know, it's quick, it's easy to see, you know, and it makes for a nice appearance when you're trying to figure out colony morphology and form and elevation and all that good stuff. Typhoid fever. So Salmonella typhi is the causative agent of typhoid fever. This has made infamous by Typhoid Mary. So Mary Malone was a cook in the 1800s. So she was actually an immigrant. She came over and worked for various wealthy families. In the course of her career, she ended up actually infecting 122 people. Of those 122 people, 5 people died. Unfortunately, because Typhoid Mary was an asymptomatic carrier, she did not fully believe that she was sick. And then there was some unethical retaining situations where Typhoid Mary was put on isolation. They obtained fecal content from her, trying to study this disease, figure out the causative agent, how it was being spread. But ultimately, she just refused to stop cooking, and so she was actually eventually sent to an island in quarantine. So typhoid fever, the bacillus typically enters in through the fecal-oral route, and then it is often sometimes spread by close contact. In the case of Typhoid Mary, the assumption is that it was fecal-oral route. If you are interested in just a bit of that information, you can click the link here. This is just a really short historical article talking about Typhoid Mary. So Typhoid Mary was an asymptomatic carrier, so she was shedding bacilli chronically from the gallbladder. So she, eventually after her death, they did isolate her gallbladder and find that that was the source of the bacilli. And so when she was cooking for the families, essentially her fecally contaminated hands and things were contaminated in the food products, and then those people were suffering from invasive diarrhea. In some cases, clearly at least 5 people had systemic septicemia and died. In 1911, there were vaccinations, and then later, around 1950, we finally have an antibiotic to treat this particular infection. Other salmonellosis, these are much less severe than that of typhoid, and so all other salmonella diseases are called enteric fevers. They cause, pretty much a basic food poisoning and gastroenteritis versus typhoid fever is causing septicemia and a much more invasive diarrhea. So you can see here the perforations and actual break through the membrane of the intestinal tract, and so you can see this swollen and pretty nastily damaged intestinal wall. And so the salmonellosis, being less severe, are still of concern. So salmonella can be transmitted through poultry, various rodents, dairy products, and things like that. We pasteurize a lot of our foods because of salmonella, reducing the likelihood of this bacteria being located there. There's various serotypes of enteritis. Some people wash their turkey, or they wash their chicken in the sink. And this is actually a really bad practice, because if you're washing your turkey or your chicken in the sink, you're splashing all of the raw juices all over the kitchen and counters. And so ultimately the important thing is to take the turkey or the chicken out of the bag and then immediately dress the turkey or chicken in however you wish to season it or fix it for the meal. Put it into the oven, and then immediately after, spray everything down with a proper amount of disinfectant. Let that disinfectant soak on there for as long as you can before scrubbing and then cleaning everything else up. That is a big contributor for the spread of salmonellas. Additionally, salmonellas are spread because of undercooked food. So if you don't cook your meat or your poultry to proper temperatures, then you're more likely to get salmonella from contaminated poultry products. So I've got a little side note here asking you why you would not want to slow diarrhea if you have salmonella poisoning. So why would you not want to go grab some Imodium or something to keep the diarrhea from continuing? So ultimately, the reason is the diarrhea is helping your body flush the bacteria out. And so you need that diarrhea to continue so that you can remove all the salmonella. Because if the salmonella persists, then it's going to continue secreting toxins and things like that are going to cause intestinal damage. So other enteric-type bacteria are that of Shigella. So Shigella has a bunch of strains. A few are listed here. And so the strains of Shigella cause a pretty crippling abdominal cramp, frequent defecation and watery stools. I included a cute little joke there. So again, it's another causative agent of diarrhea. The difference with Shigella, however, is that Shigella is primarily a human pathogen. So we're picking up Shigella not from poultry, not from animal sources, although it does appear to also be able to infect apes. But Shigella is usually transmitted from person to person or through a nosocomial situation. So the virulence factor for Shigella is Shiga toxin. So Shiga toxin is produced both by Shigella and it is also produced by E. coli. I'm going to take you back there. So enterohemorrhagic E. coli also produces Shiga toxin. So don't forget that part as well. And so they share the Shiga toxin. So likely at some point in the distant past, Shigella or E. coli shared that Shiga toxin, and they now both possess that virulence factor on a plasmid. And so the site of infection, of course, is still our large intestines. We have damage and things due to endotoxins and enterotoxins causing some nasty diarrhea. Yersinia pestis is the causative agent of the plague. It's some very, very tiny gram-negative rods that do have capsules, so it's resistant to phagocytosis, which is good because it's going to hide out in those phagocytes and persist intracellularly. This bacteria produces the virulence factor coagulase. So coagulase is the clotting factor, so it will actually block the entry of other phagocytes and things like that by clotting blood on purpose. So it will kind of use the clotting feature against you. And then it has endotoxins and, of course, marine toxin. Marine toxin makes this particular bacteria very virulent to mice, but marine toxin makes this bacteria able to survive in fleas. Fleas are the vector for Yersinia pestis. So the sacrificing of mice is beneficial in the sense that the flea vectors remain very ravenous because the coagulase will clot the blood. And so when the coagulase clots the blood, this flea becomes ravenous because it's not ever getting sustenance. And so the flea continues to feed repeatedly on several mice, several other species, or even humans. And so the flea then can transmit the disease to pretty much large quantities of populations. Shown here, you've got a mass grave from the Black Death. The Black Death, or the bubonic plague, was extraordinarily devastating. So it's probably right behind the influenza outbreak, the 1918 flu pandemic. In this case, 25 million people died, so nearly, a little over half of the European population at the time. They had to put people in mass graves and burn them to keep them from disseminating infection because the mortality rate was so devastating in this particular outbreak. So the infection cycle of Yersinia pestis is related to that flea. So the flea is ravenous, it's got the bacillus bacteria in it, and then the intermediate hosts are either mice, chipmunks, could be a ground squirrel, all these guys. And so the flea can either be a direct source of the infection, or it can be a vector for the infection between the final host and the intermediate host. In humans, we can have bubonic plague, pneumonic plague. The most severe form is the pneumonic plague. Pneumonic plague, because it is so highly infectious, the transmission of aerosolized droplets, so it's going to be fatal without treatment. The bubonic plague is the formation of a bubo. So buboes are swollen lymph nodes, and so you can see a pretty significant bubo here. And then septicemic plague. So this septicemic means that the bacilli has gone systemic. It's in the blood. It's actively replicating. And that is what is responsible for what we now know as the Black Death. So the septicemic plague is this black purpura where the coagulation and hemorrhaging of these cutaneous tissues. So think of if you've ever really severely stubbed your toe, and the toe or the area underneath the fingernail bed, turns black. Well, that's essentially the same kind of thing that's happening here, but it's happening because of the bacteria. And plague, because of its virulence and the ability for it to be a pneumonic form, allows it to be a really big bioterrorism threat. So we're a bit concerned about keeping all the plague locked up in the proper refrigerators or freezers of the researchers who are actually doing those sorts of studies. And then the last bacteria that we'll discuss is that of Haemophilus. So Haemophilus is a gram-negative pleomorphic rod. And so Haemophilus causes things like otitis media, bacterial meningitis epiglottis, but distinctly it's a big causative agent of otitis media in children. And it's partly because the eustachian tube. Children, as they're growing, the eustachian tube is first horizontal, and then as you move toward adulthood, the eustachian tube then is vertical and drains. And so a horizontal eustachian tube allows the Haemophilus influenza bacteria to colonize, create a biofilm, and then, of course, cause disease. We, of course, now treat Haemophilus with the Hib vaccine, and so the cases of otitis media and these other diseases have gone down pretty significantly, and you can see that information here. And then the Haemophilus aegyptus is the causative agent of pink eye, and so you can see this is a bacterial pink eye, and you can tell that it's a bacterial pink eye based on the really bright purpura underneath the eyelid. So you can see that the whole entire eyelid, viral pink eye, has a slightly different presentation. So prevention for all of these diseases is wash your hands, wash your hands, wash your hands, clean your food, clean your food, clean your food, and cook your food fully. So that's kind of ridiculous, but really, wash your hands, clean your food, cook your food. These sorts of very logical approaches can reduce the spread of these sorts of diseases pretty readily. And then the last concept checked is that of a bubo. So a bubo of bubonic plague is what exactly? Ulcer where the flea bite had occurred, granuloma in the skin, enlarged lymph node, infected sebaceous gland. So just as a quick reminder, the bubo was in the axillary region. It was very, very big and round and probably close to the size of a tennis ball. And it was indeed an enlarged lymph node.